occasionally have these slides of the sort of thing I was just showing of what I think of as biology appreciation slides. Would anybody be interested in my having those available on Icon for your own downloads? If you're interested in that, could you raise your hands? That's easy. All right. If you're shy about saying it, come by after class and let me know. Do people have any questions about the material we've talked about so far? Talking about some of the kinds and varieties of nucleotide of DNA repair. Questions? Comments? I believe this was the last slide that we were talking about. The idea that one particular form of one of the diseases that gave us insight into the genetic basis of DNA repair is a variant of Xeroderma pigmentosa in a very special kind of DNA polymerase called DNA polymerase eta, again counting in Greek, alpha, beta, gamma, delta, zeta, eta. And polymerase eta has the unusual property that when it sees a thymine dimer where it's been chemically cross-linked by UV, it can in fact place an A across from it correctly. When it sees an 8-oxoguanine, it can correctly put a C across from it. So it can replicate even though a base is damaged. And this is because it has the active site where stuff comes in that's enough more flexible to accommodate these differences. And in Xeroderma pigmentosa with this particular variant form, because this enzyme is missing, the damage due to UV tends to accumulate, and it shows up specifically as skin cancer because that's the prevalent form that it's correcting. But this introduces us to the concept of translesion DNA polymerases. So this is a group of several different DNA polymerases that are capable of making DNA in the context of damaged DNA. So normally, base excision repair or nucleotide excision repair will detect and correct damage that's been done. But in some cases, it fails to. And in the cases where it fails to, some of these translesion DNA polymerases are capable of picking up the job and carrying on with it, each of them having different kinds of damage that it's better at repairing than others. The translesion DNA polymerases are not as accurate as the bulk DNA polymerase. Lynn talked a bit about how very accurate DNA polymerase is, how it has a proofreading capacity to go back and actually catch its own mistakes and correct it. But there is also room for doing something well enough, and that's what the translesion polymerases do, is that they allow there to be at least some kind of DNA there and allow for the ongoing replication of it in the context of the damage so that you have a chance at least to carry on. It's a balance. Genetic studies on E. coli have shown that you can have a certain ongoing rate of mutations, and you can select for higher and lower rates of mutation. But in both cases, the cells grow more slowly. And it makes sense they grow more slowly when they're making more mistakes because they're making mistakes and they're cribbing on themselves. It may not be as obvious that they grow more slowly when they're more accurate because they have to invest more time and more effort to correct all the mistakes. So there's a balance to be reached. So in addition to things where bases are damaged or a single base is fallen off hydrolytically or something like that, it can happen that there are actually a gap in the DNA where there's actually a complete multiple base break. And these are hard to keep from happening altogether. So they can happen, and since they can happen, they do happen. But they're not good things. When DNA polymerases come to them, they're going to not want to go through and make more DNA at that spot. So gaps in the DNA inhibit replication. And there has to be some way to cure the gaps in order to get back on with the business of living. And this brings us to the process called recombination. And recombination is a bag of tricks that allows cells to overcome the presence of gaps in their DNA in ways that allow, for the most part, to be faithful to the original sequences. And we're going to see that there are ways of approaching both single-stranded gaps and also double-stranded gaps in order to try and repair these things. Recombination processes, we can tell, are extremely important because they're highly conserved, both among prokaryotes and eukaryotes. This is part of the toolkit that cells don't let go of. They need some way to maintain DNA at the highest feasible level of accuracy. And this is one way that they can maintain the integrity of DNA. So let's take a little look at single-strand breaks, which are the first and easiest things to deal with. So here we have at the top a replication fork. And here's the leading strand rolling along, and here's part of the lagging strand that's been, I'm sorry, it's the other way around. This would be the leading strand, that's the lagging strand. But they've come up upon a gap. They detect a gap, and the gap in that spot is going to prevent replication through that region. Helicase is able to unwind the new DNA at that spot. So this part is new DNA, and that part is new DNA. And when helicase unwinds that little bit, what you end up with is DNA that looks like this, with an overhang from the old DNA and the new DNA that's only gotten that far. This free 3' end here is able to now, in what they call, invade the double-stranded DNA. And when it invades there, it displaces the other strand. And so now you have it able to pick up the information from that strand of DNA. Ultimately, this crossover is going to have to be resolved. That is, there's protein machinery that allows the DNA to become untwisted at that spot. And after resolution, this is the structure of the molecule at that time. And it's still color-coded, so you can see which parts were where. So in the original molecule here that got stalled, you have a strand of DNA that runs to there. There's a gap at that spot and continues like that. And its complement was continuous. Here it's shown as red all the way through. If you look at the resolved and partially repaired structure, you can see that the original strand that had a gap still has a gap. But it has a complementary strand across from that gap, just as it had up here. And the other strand is an integrated, complete, nothing missing thing, where this bit was able to be copied in by borrowing it, so to speak. And this gap here can be then completed by polymerase and ligase because it's not in the middle of the whole event of replication. Things get a little messier if both strands have broken. And so if we have a situation where we have a two-strand break like this, one way of approaching this to resolve it is for a nuclease to come in and nibble off some of this DNA, nibble off some of that DNA, and now we have two free flapping 3' ends. And those free 3' ends can invade homologous DNA. So homologous DNA would be another copy of that DNA that has the same information as yours, as the strand you're looking at. In us, because we're diploid, we have homologous copies for everything. If you were haploid, like some of the fern plants and stuff, you wouldn't have a homologous copy around, so you'd sort of be out of luck. So these free 3' ends need to have a nearby homologous DNA that they can invade. And when they do, they can then pick up the missing sequence by borrowing it or by copying off of the homologous copy. The structure that it looks like at that point looks rather complicated. And the resolution of this sort of crossed over thing, again, takes protein machinery to allow it to untwist and resolve itself into a structure like this. This would be a gap that would need to be completed with ligase after completion and so on. But you can see that by doing this, you can borrow the sequence you need to complete the job of copying that DNA. So recombination is a way of dealing with these breaks in DNA. It is something that may have to be done during replication, but the genes that are used for this are expressed all the time. This is not something that can be left lying around and sort of get to it on April 15th with your tax returns. The genes for this are constitutively expressed, meaning they're expressed all the time. And one of the important members of this is a protein called REC-A, or recombination A. And it can bind to double-stranded DNA. If you look at this picture over here, you can see in the middle of it in red a core that is a DNA helix, and this kind of silicone-looking stuff that's been coating around it is the REC-A protein. And REC-A, as you can see here, actually makes almost like a polymer over it. That is, the REC-A lies down in multiple copies on it, and it can untwist the DNA by using ATP to drive that process. And doing that, it makes it easier for these free 3' ends to get in, to invade, and base pair, and pick up the information from the complementary stand. We have assigned that REC-A is an extremely important protein because people who are lacking REC-A have a genetic disease called ataxia telangiectasia. And so this is, again, one of these genetic windows that tell us just how important this protein is to ongoing, healthy, regular life. Excuse me. When you have a two-strand break, in addition to this recombination approach that we just talked about, there is the potential for simply joining blunt ends together. If you do an end join like that, where you just join a blunt end together, you don't need to have a homologous molecule that's intact to invade and repair from. You match up a 5' to a 3' end and put them together and then ligate them. You can see that there's real potential for difficulty here if you match it up with the wrong thing. When you do an end joining between two things that were not originally part of the same gene, what you've done is to generate a new gene that's part A and part B. It may not be very well put together for that. And so end joining does have an increased potential for being mutagenic compared to some of the other approaches. We opened our discussion of DNA repair with Deinococcus. Deinococcus is great at doing this. You may remember the picture we had at the very beginning of something that looked like confetti, where the DNA was chopped up into little pieces. And Deinococcus is so good at doing this that it can survive, as I said, thousands of times more radiation than we can. They think, in part, that is because you notice here it says sort of magically alignment, like pick up the right end, sort of then a miracle happens stage. Deinococcus seems to have multiple copies of its genes that are kind of kept in a barrel or a braid so that they tend to be near each other. So that may be part of why it's so good at it. So we've talked about some of the varieties of DNA damage, how we can lose just the base from the nucleotide. We can actually lose the base and the sugar part. We can have chemical modifications. We can have an entire gap and so on. We've talked about some of the mechanisms for repairing this that are, in each case, relevant to the kinds of the damage. And we've seen that these processes are very important because damage to the genes for those processes shows up as actual serious genetic diseases. So what are some of the genes that are damaged in cancer? what are the specific targets that especially show up in cells that have gone astray in the form of cancer. And we can talk about them in a couple of broad ways. We can talk about cell cycle progression. That is, cells have an orderly way of doing their work on a regular basis. And we're going to look at the cell cycle. And that is regulated through proteins called cyclins, which is a logical name for something dealing with the cell cycle. There are also what can be called quality control checkpoints, places in the cell cycle where the cell can kind of stand back and say, are things going as well as I think they are or not, and pick up on that, often through signaling pathways. So let's take a little peek at the cell cycle. This is just a schematic cell cycle, a one-size-fits-all. Cells differ in the relative sizes of these phases. There are some organisms that have a very large variation on this. I studied an organism for a long time that had a very long, like multi-day growth phase, and then went synthesis, mitosis, synthesis, mitosis, synthesis, mitosis, like as if it were a sea urchin egg or something. But this is a good general model, and it's a good one to compare others to. There's a first phase called G1. Here they're calling it growth 1. And in fact, a lot of the growth happens during the G phases. It was originally called the gap phase. It's the gap between mitosis and synthesis. But you can think of it as growth if you want, G1. G1 is followed by S, and S is for synthesis, but it is specifically the synthesis of DNA. There's a lot of other kinds of synthesis that goes on in G1, RNA synthesis, protein synthesis. S phase is for DNA synthesis. There's a second G phase, G2, or the gap between synthesis and mitosis, or growth between synthesis and mitosis. And then the end of the cell cycle is mitosis, being able to separate your DNA into two equal exact packages, being able to separate your cytoplasm into two working bundles, and then go back and do it all over again. So those are the stages in which we describe the cell cycle itself. If we observe, we will notice that there are some proteins that rise and fall with these phases of the cell cycle. So for example, if you look at the abundance of cyclin D, it's undetectable early in G1. It starts to rise to a peak in late G1, and it's almost entirely gone when synthesis starts. A related cyclin starts a bit later, peaks a bit later, but extends partway into S phase. Cyclin A is basically coterminous with the S phase. Cyclin A or B might stretch over the next part as well. So these are proteins whose abundance is very tightly tied to the phases of the cell cycle as defined by things like DNA synthesis or mitosis. It turns out that cyclins are in fact part of controlling the cell cycle. They play a significant role in controlling the shift between G1 and S, or S and G2. And we're going to see that there are kinases, protein kinases, that are themselves regulated through cyclins that then go and interact with signaling pathways. I don't expect you to memorize any of these numbers, okay? I just wanted to show you some real data as opposed to a generic one. This is a yeast cell cycle, and you can see here the G1 phase, S, a small sliver of G2 and an M. And you can see some of the kinds of things that are involved in the cell cycle. So for instance, thymidylate synthase. Well, obviously you need thymidylate DTTP to make more DNA. So it's logical that that would be a cell cycle dependent enzyme. Here's DNA ligase as one of them. So those are all things that are logically part of a cell cycle. And they're showing you the sequence of some of the events here of initiating DNA synthesis, the bulk of the DNA synthesis, beginning, in the case of yeast, you actually begin some of the work of dividing the nucleus while you're making more DNA, so that the idea of S and M are a little bit overlapping compared to mammalian cells because the spindles actually start forming while you're making DNA. And then you have other morphological features like growing a bud or cells separating and so on. Another concept that fits into the notion of cell cycles is the idea of cell cycle checkpoints. That there are certain parts of the cell cycle in which there's machinery available to stop and look and answer key questions about the status of the cell. And so in late G1, there's a checkpoint called start, meaning you have to get past this point with a yes answer if you want to start making DNA, if you want to go into S phase. And some of the questions that feed into the start checkpoint are, is the cell big enough? Is the environment favorable? You know, do you have enough amino acids around to make more stuff and so on? If you pass the start, you can proceed then into S phase, replicate your DNA, get into G phase, G2 phase. G2 has a checkpoint also, a checkpoint at the end of it, which is concerned about whether or not to enter mitosis. And so some of the questions that the machinery is asking is all of the DNA replicated? Is it still an okay place to live? You know, is the environment still okay? Is the cell big enough? And so these are other features that have to be finished. And if all of those get yes answers, you then start into mitosis, divide your chromosomes, start dividing your cells. And there's a metaphase checkpoint to look at whether you should finish up the whole process. If all of the chromosomes are correctly aligned, that's a sign that it's time to go ahead and complete the process of mitosis. If they're not all there yet, the cell can detect that and wait for a while. And maybe it'll wait there for quite a while until everything is right in place to finish up the job. So some of the things that can feed into these as checkpoints would be the growth signals. That would be a sign about the environment being favorable. DNA damage might be a negative signal in terms of DNA replication, telomere length. If the answers to these things are no instead of yes, the way they need to be, either the cell cycle stops at that point and the cell tries to get its act in gear and get everything in place, or if it's serious enough, it can trigger a process called apoptosis. And we're going to take a little look at what apoptosis looks like. It's a very ugly picture indeed. Very ugly indeed. Here's apoptosis. Although I guess actually when you color it lavender, it's not so ugly. Apoptosis is a process that's been well appreciated in the last 10 to 15 years. And it's the idea that cells have machinery in place all the time, ready to commit suicide. That there's a program within the cells so that the cell will die. And the stuff that's inside the cell gets wrapped up into membranes and is eaten up by macrophages so it can get recycled. There may be external signals that will trigger apoptosis within the cell itself. If there's the wrong kind of damage, if it stays at a checkpoint too long, it will trigger it. And part of how it's accomplished is through caspases. And caspases are calcium-activated proteases. Now, normally, the calcium content of the cell is quite low. The free calcium level is quite low. But if your mitochondria start to leak, the calcium levels will rise, and that provides the calcium through which the caspases can then start basically breaking down everything in sight. And it's something that once it gets started, it goes more and more so. There gets to be a no point of no return beyond which what's going to happen to the cell is it ends up looking like this. These little bubbles on the surface of this cell are called blebs. And blebs are, in general, a bad sign on your cells. And they are places where the membrane has come separated from the cytoskeleton. And in blebs, and for the most part only in blebs, membrane proteins are free to diffuse. They're one of the very few places where the membrane proteins still or can diffuse freely. Any questions? Okay. So Now, I'm not going to try to walk through each of these cell cycles. What I would like you to take away from it is that the signaling pathways in the cell are, first of all, there's multiple signaling pathways that can integrate information from the outside about the environmental status and whether it's time to grow, and that these pathways are not completely isolated from each other, that they tend to talk to each other to some extent. These cell signaling pathways include kinases, that is, proteins that phosphorylate other things, as we saw during the regulation of metabolism. They include regulated phosphatases that will take off those phosphates. So the old, if there's an on signal, there's an off signal, and other components as well. And you end up with not just a linear pathway, you end up with more like a network, something that is cross-linked enough that you're not just looking at a straight line of behavior. That's good to the extent that it makes it somewhat redundant. You would like cells to be able to cope with a lot of variety, and redundancy is one way to do that. But it also makes it harder to pin down exactly which component affected which pathway. So you may know that, for example, cyclin D is a key part of the cell cycle. You may not find it as easy to tell back which of the pathways it was definitively involved with. We do know that defects in these pathways tend to be linked to cancer. That is, defects in the cell signaling pathways are one of the things we frequently see in cancers. I mentioned that there's a DNA damage checkpoint, and that the cell won't go past that point if it thinks that the DNA is not intact and happy and everything. And it works in general terms, something like this. You have a DNA molecule that's somehow or other has swallowed a starfish or been damaged some other way. And there's a sensor protein. There's a protein that is capable of detecting and binding to that part of the DNA that has that kind of chemical damage. That sensor protein will trigger the activity of kinases or other transducers to let them know, I'm sitting down on a piece of damage. They then can activate a number of other things that will help repair the damage and tie the progression of the cell cycle to that repair. So the kinds of things typically that these transducers can interact with are the repair enzymes that we've talked about, transcriptional regulators that can turn on or turn off things that you want going the moment, the cell cycle control proteins we've been talking about, or if it's a bad enough kind of damage, apoptosis. Overall, what it's going to do is it's at least going to stop that cell cycle until the damage is repaired. It may decide that it's not worth fixing and trigger apoptosis. There's an important one called ATM, which is a protein kinase, that is one of the important kinases in this cascade. The ATM itself has as its targets two other proteins that recur very frequently in discussions of cancer. One of them is called BRCA1 and one's called P53. These are very boring names. I don't necessarily expect you to memorize them per se. But BRCA1 got its name for breast cancer. This was one of the first genes picked up in association with familial patterns of breast cancer. And P53 is simply a 53 kilodalton protein. Biochemists sometimes have very little imagination. You're not going to tell my department chair I said that. So let's look a little bit at what BRCA1 and BRCA2 are. This is a partial picture of the structure of BRCA. One or the other of these being mutated can raise your risks to 70% of having breast cancer. There are families in which BRCA genes are present, mutant ones are present, where some of the women have mastectomies prophylactically in their 20s rather than face the risk. I can't begin to fathom feeling that that's a choice I need to make. They are targets, as I mentioned, for this protein ATM. And if they are phosphorylated by ATM, they may participate as a scaffold in the whole process of identifying damaged DNA and helping to repair it, helping to recruit recombination enzyme proteins, and so on. In cells that are missing BRCA1, this kind of cell cycle control and repair are not very functional. And so when damage occurs, it's hard to get it fixed. One thought is that BRCA1 might perhaps bind to double-stranded DNA breaks. In other words, you remember we were talking about how much harder two-strand breaks are to fix than one-strand breaks, and one of the possible roles for BRCA1 is to hold those two things together until the cavalry arrives. Another thing that can contribute to cancer formation is losing control of the cell cycle itself. If you can't manage the rate at which you progress through it, if you can't manage to listen to the checkpoints, that can be one of the stages on that multi-step pathway towards turning into cancer. And one of the control proteins that turns up frequently is a protein called RB, which is short for retinoblastoma. And RB protein was first identified through children who have a genetic form of retinoblastoma, which is, as you might guess, a cancer of the retina. And so these are children who had defects in this gene. The normal activity of RB is to keep cells in G1, to hold them in G1 for long enough to complete all the work that they're supposed to do and not go ahead unless there's a reason to go ahead. Many of the cells in the body should not divide. If all of your cells were dividing all the time, you'd be, bigger than Brobdingnac or somebody like that. You want to be the right size. And so most cells are not going to divide. And part of that not dividing is the work that retinoblastoma has of holding things in G1 so they don't go ahead unless it's necessary. One of the things that RB does is bind to a transcription factor, E2F. E2F is needed to make the cyclins for S. And so if RB activity is high, E2F activity down here is low. And RB activity has to fall for E2 activity to rise. We find in many cancers mutations of one kind or another in the RB protein. It takes a double hit in it to cause the eye cancers, and I don't know the biology of the eye well enough to understand why the double hit shows up, especially as retinal cancers. But I think this diagram does show you this idea of complementary activity, that you need one kind of thing going in one phase and then a different thing going in the next phase. Another protein that turns up in many cancers as being important is P53. Again, P53 has an extremely boring name. It's just 53 kilodaltons. For such an exciting protein, it has a really boring name. It's a tumor suppressor gene. So its presence tends to damp down the development of tumors. And it is mutated in a good half of all tumors. In a normal cell, the amount of P53 is controlled by how fast it's degraded. And ATM, if you remember that gene from back there when we were talking about cell cycle control and DNA regulation, can phosphorylate P53 and make it last longer. And in that case, the P53 will delay the cell from moving forward into the cell cycle. It'll hold it back and get the time that's needed for the DNA repair machinery to get there, to activate the enzymes and get things fixed up again before they go on to the rest of the cell cycle. It also has a positive effect on increasing the synthesis of deoxynucleotide triphosphates, which obviously you would need in order for DNA repair. I'm not going to ask you to memorize this path, but I think what I'd like you to take away from it is that this one protein, P53, can have multiple targets as a way to accomplish its goal. It sometimes has been called the guardian of the genome, which sounds real glamorous. But it's out there watching for damage and trying to protect the genome. So if there's DNA damage, P53 will do stuff, and it'll do several kinds of stuff depending on the levels of damage and what's needed. If DNA damage is extensive, P53 can act on BAX, which is a key part of the pathway to apoptosis. And this sort of pathway for P53 is saying, this is past anything that's worth repairing. Let's not invest the effort to try and fix it. Die. Just die and go away. And so that's one possible outcome. If the damage is more moderate, P53 can then act in a couple of ways to either buy the time for repair or to damp down the things that would make the situation worse. So it can act through P51 to stop the cell cycle, and it can act on other genes that are part of the expression pathway for the oncogenes that we've talked about, that are things that favor tumor formation. So it's pretty central to cancer and prevention of it. This is a diagram that shows you sort of the consequences of P53 and what happens if it's missing. So here we have at the top two different cells. Here's a normal cell that has a normal amount of P53 that's functional. Its companion over here is going to be a cell that has either a mutated or a gone P53. And both of them are going to be exposed to something that could damage its DNA. So that could be ionizing radiation, which creates ions right inside the cell when the radiation is absorbed. They could be carcinogens that are going to intercalate into the DNA and make mismatched sequences. They could be mutagens that chemically change the DNA, whatever. They damage the DNA. Somehow or other, they've damaged the DNA. So now there's DNA damage in the cell. The P53 is around, it's activated, it binds to that DNA. And at this point, several things get turned on. We turn on a protein that can stop the cell in G1. We turn on proteins that are involved in DNA repair. Or if there's a lot of them, we can turn on apoptosis and just call it a bad job and go die. If we arrest and we turn on DNA repair, there's a decent chance that we can, in fact, correct the DNA and go back to normal life. Well, what about the mutant cell that doesn't have P53? It's exposed to the same things. It has DNA damage. So it has now places where the DNA in it is not what it should be. But in the absence of P53, this guardian of the genome, it doesn't turn on the DNA repair. It doesn't stop the cell cycle. And it can't die through apoptosis. And so these DNA damage persists and leads to mutant cells that now have other mutations, other kinds of errors in their DNA. And for some of those mutant cells, there'll be ones that are quite compatible with living, and in fact, they may grow faster than they grew before or grow in a more uncontrolled way. And so this is one of the reasons why damage or loss of P53 is so common in cancer cells. Take this as a very overview slide. I'm not going to ask you to read all the elements of it. It's meant to sort of draw your attention to the fact that cells integrate information from a large variety of sources about whether or not to grow, whether or not to divide. There are some that are favoring growth. Here's growth factors. There are some things that are against growth. There's antiproliferative. There are external things that turn off growth. And P53 right here, as a DNA damage sensor, is at a place where it can interact with several components of that cycle. And RB, which is up here, is also placed, but to interact with different components of that cell cycle, or cell network almost. So we need to be aware of the fact that our bodies are looking out all the time for things going wrong. There's surveillance going on all the time to try to detect errors at an early point. That surveillance is an ongoing process. It may take the form of genes that are turned on that shouldn't be. It may take the form of DNA damage we've been talking about. Any of these things that are triggered by the tumor-generous sensors can trigger then several things. Retinoblastoma, if it's activated, can turn the cell cycle down or off so you don't keep going. P53 can also turn the cell cycle down. It can kill the cell. Other pathways as well that we haven't even talked about. But this means that generally you're going to need both RB and P53 damage to allow full tumor development or something equivalent to it. And then I believe I've mentioned, although I'm not sure I have, the fact that angiogenesis is also a key component of this. That you could have a small mass of cells that has, has a dead RB, it has a dead P53, it's got mutations out the wazoo, it's aneuploid, it's ready to go. If it doesn't have a blood supply, it can't grow very big because it'll have to get everything by diffusion from the fluids of the body. And that's about 1 millimeter is about as big as it's going to get. So at that point in carcinogenesis or tumor development, the little mass either will or won't acquire the ability to recruit blood vessels. And it's just a random thing whether or not it does or doesn't acquire that. But once it can start to recruit blood vessels, and you can see in this little diagram, these little green dots are the things that are attracting the blood vessels, you can end up with a tumor that gets, can recruit those blood vessels to invade it and supply it with all the nutrients that it needs. Unfortunately, the same blood supply that supplies it with the nutrients it needs also provides a track back into the body where metastatic cells can enter the bloodstream and perhaps find a home somewhere else, which introduces us to the notion of metastasis. Doctors sometimes have kind of black humor, and so the joke goes that in the oncology ward, the Mets always win. Mets is the nickname for metastases. As an ex-New Yorker who used to root for the Mets, I'm very ambivalent about that joke. So what is a metastasis? Well, the word means standing, stasis is standing, and meta, over, beyond. So these are cells that have gone out and taken up places elsewhere. If you have a situation like this, this is a case where the cells are disordered, they don't have good growth control, they don't have good morphology, but they haven't yet figured out a way to recruit a blood supply. We call that carcinoma in situ. In other words, a carcinoma in place, right there. Within this carcinoma in situ, one cell might acquire the ability to release the angiogenic factors that are needed to recruit a blood supply. And they've colored that one cell as black. And so it's going to start growing better because it's recruited a blood supply. Now, you have now this microtumor that has a local blood supply, and a couple of things can happen at this point. At one point, you can have just a small breach in the surface where a few cells happen to fall out and maybe find a new home. Of a more serious consideration is that the part which has the blood supply and is vascularized, that these cells can take a trip out through that blood supply and then land somewhere else in the body in a capillary bed and stay there because the capillary bed is a fairly tight place and it can't get any further. And then those colonies that are seeded elsewhere in the body are metastases. My husband's a pathologist, and he's very fond of this cartoon. I don't know if you can read it on your handouts. It says, the guy up on the top of this says, "It's a mammoth." And at the bottom it says, "Early microscope." Pathologists have a strange sense of humor. But they can be very nice anyway. So we've talked about cancer and some of its characteristics, and these are things that we're going to see in all of them. genome instability, the idea that the normal complement of genes that, most of our cells happily go through using again and again is disrupted in some way. And so now the number of chromosomes may vary. There may be breaks in the chromosomes. The integrity of the genome itself starts to go away. Cancer cells no longer require external growth signals. So all the normal tissues of your body have some spectrum of growth signals that they can detect and respond to. Cancer has gotten beyond that. It's like, a really distressing 15-year-old or something. They've stopped listening to growth inhibition. Normal tissues have both chemical signals and physical patterns that will signal to that cell, stop growing. Cancer cells have learned to ignore those signals. They found a way to evade apoptosis, the programmed death that should be there as sort of the last insurance policy. They found a way to bypass them. They have no limit to how often they can divide and replicate themselves. And that's a combination of things. They don't senesce. That is, there's a program of chemical changes that happens towards the end of a cell lineage called senescence. And they also don't differentiate. So that, for instance, the stem cells in your gut will divide many, many, many times, but their progeny will differentiate. If they don't differentiate and just stay on being stem cells, then you develop a tumor. There has to be, as I mentioned, the need for angiogenesis, and then as the grade of the cancer progresses, you have invasion and even metastasis. I'd like to share just a little bit, if we have time. I think we have time, yeah, about one particular cancer, because it's a cancer that has fairly broad prevalence and significance, which is cervical cancer. And the human papillomaviruses are the cause of cervical cancer. They invade only squamous epithelia, which are the very kind of layered cells, tissues that have flat layers of cells that stack up, or squamous epithelia. Many of you have inquired one or another kind of an HPV because warts are caused by HPV. And there's a very large variety of them. There's over 100 known strains of HPV. Some have called HPV a nearly perfect virus because so many people have it for so long, it almost never kills its host, and so on. So HPV viruses are very smart that way. However, there are a subset of the viruses that are very bad players, don't work well with others, and that kind of thing. And that subset are routinely found in the invasive and the metastatic cervical cancers. That group of HPVs are called high risk because of that, and they are transmitted sexually, although people may not know they have it at all because there may not be any immediate sign of the HPV infection. One of the other things that has come out of these studies is that you have to have an HPV virus, a papillomavirus, to have cervical cancer, but that's not enough. That is, just being infected is not enough. There's other things going on. And it will progress over time. 2% of the cancer deaths in the United States are due to this virus. Tragically, 25% of the cancer deaths in Mexico are due to this virus. And this reflects the fact that Pap smears, in fact, are a very effective way of catching cancers early. They don't prevent the cancers, but they catch them at a time when they are treatable. And we've uncovered what it is in HPV that does this. There's two proteins in particular that are extremely vicious, whose nasty work makes sense in terms of the things we've been talking about so far, and this is one of my reasons for bringing up HPV. The E6 protein binds to P53, our guardian of the genome guy, and causes it to be degraded. E7 binds to RB, our guy that could control cell growth and stuff, and inactivates it. So these two viral proteins take out two of the major protectors for the normal cell. And then in addition to that, the virus can induce this genomic instability we've talked about and prolong the lifespan. All of these things together obviously make it easier for a cancer to develop. To give you a little bit of a sense of time scale, none of these things happens instantaneously. So this is a time course from the initial infection here with HPV, and the cells are going to lose RB and P53 early on, but it might take 13 years before the lowest grade kind of carcinoma is detected. And these are cells that are immortal, that are beginning to show angiogenesis and some factors. And it'll take more time still before these carcinoma in situs can invade and metastasize. And I'll let you look at that picture by yourself. I think it's fairly self-explanatory. That actually works very well, doesn't it? Putting it up on the screen like that.